Is Paula, and today we're going to be learning about a technique that's currently used with some cutting-edge research, and it's called PCR. We're going to be doing a couple of games, we're going to watch a video, and we're going to learn about how the technique works. But before we get into that, we're going to do an icebreaker. We're going to go around the room and everyone say your name, say what your favorite subject is in class, and say what your favorite board game is or your favorite video game. So pause this video and let's do an icebreaker. Okay, so to go over the basics, PCR stands for polymerase chain reaction. So this is a technique that starts with a very tiny sample of DNA, and maybe an example is some cells from a woolly mammoth. We're going to take those and we're going to amplify them, and that's going to allow us to study this organism in more detail. So there's a lot of different uses for this. It can let us study viruses or bacteria. We can identify an individual maybe, and more things that we'll get to talking about later. So these are the steps to PCR. First, we start with our sample. And within that DNA strand, we're going to zero in on a gene or a target DNA segment that we're interested in. So we're going to throw that into the PCR, and that is going to make millions and billions of copies for us. So with those copies, now we're not working with some very small segment and only a single segment maybe. Now we have enough DNA that we can do any amount of researching we want to do or genetic characterization that we want to do. So the step with the thermocycler, there's a couple of things that we're going to need in order to do this. So this is how PCR actually works if we're in a lab doing it ourselves. So we have our small piece of DNA, and it goes into a really tiny tube. And with that DNA, we're also going to need to put in that tiny tube primers, our reverse and our forward primers, which we can talk about a little bit later. We're going to need nucleotide bases because remember, we're taking this one small sample and we're going to end up with millions of the same DNA. So it's going to need some bases to work with, and we'll talk about that. And lastly, you're going to need a polymerase enzyme. You're going to need an enzyme to do all of the work of constructing all of these strands of DNA for us. So this enzyme is a specific enzyme. It's one that can stand a lot of heat because our PCR is going to get very hot. It's going to be breaking apart our DNA to allow us to build more pieces of DNA. But let's get into that part a little bit more. Polymerase chain reaction is a method widely used to rapidly make millions and billions of copies of a specific DNA sample. It starts with selecting an interesting portion or target region of double-stranded DNA. The first step in one cycle of PCR is denaturation. The DNA must be heated to separate the double strands into two single strands. Next, the annealing step begins. The primers will place at both ends of our target DNA to provide a starting point for our new DNA formation and signal our Taq DNA polymerase. Now our heat-resistant enzyme will place and provide a starting point for the new DNA formation. Now our DNA is extending again. Once again, our DNA is denatured. Then the annealing step, we're putting down our primers. And lastly is the extension step. This is the third step. As the DNA polymerase move along the single-strand DNA, the nucleotides added to the mixture will place to their corresponding base pairs. Let's briefly go over base pairing. There's 4 bases in DNA: adenine, cytosine, guanine, and thymine. The 2 strands are held together by hydrogen bonds between the bases of adenine, forming a base pair with thymine, and cytosine forming a base pair with guanine. Now that the 3 steps are done and 1 cycle is complete, the process can begin again until we end up with millions and billions of copies. What do the black dots of clay represent? Ready? 3, 2, 1. Okay, so what step is being demonstrated here? Ready? 3, 2, 1. For our last question, you might want to count the number of strands that are here and ask yourself, are these single strands or double strands? Okay, awesome. So hopefully you got most of those answers right. So if we run our PCR, we have to have a way to look at it. If we're trying to identify an individual or we want to study some bacteria, we have to have a way to look at the results. So let's talk about that. Okay, so let's do an activity to learn how to read the results of our PCR test. So in Huntington's disease, there's a region of the genome that's made larger and larger with each generation. The larger the region is, the more likely that individual is to get the disease. So can you guess, by looking at the image on the right, who, based on just that information, who is the grandfather and who is the grandson? So the grandson would be the orange DNA, right? He's had the gene passed down to him through generations, so his region is going to be larger, and his likelihood of having Huntington's disease is going to be greater. So if we run in our PCR DNA from both this grandson and this grandfather, and we run it on a gel electrophoresis. So the way that a gel electrophoresis works is we take the DNA out of those tiny little tubes, we put them into the wells, which are the empty rectangles. These are the empty boxes. And our gel electrophoresis is going to have an electrical field run over it. There's going to be a positive end on the bottom, and DNA is negatively charged. So the DNA is going to move. The longer that you run your gel electrophoresis, you're going to plug a voltage into it, it's going to run down to the bottom of the gel. The lighter the DNA is, the faster it's going to run. So this is going to tell us the differences in size of DNA. So based on that, who do we think is DNA number 1? So we would think this is the grandfather, right? Because DNA number 1 is running faster down to the bottom of the gel than DNA number 2. And we can also read on the side here that it only has 200 base pairs, and DNA number 2 has closer to 300 base pairs. So this should give us a good idea of how to read a gel electrophoresis, which we're going to need for our activity. Okay, so individually, or maybe we can group up in teams. If you want to pause this video now to come up with your groups. Red finds a dead body in the storage. So clearly there's an imposter among us. No one is seen going down a vent, but you have DNA from the imposter. So your crew calls for a vote. We all vote and we all agree everybody is going to contribute their DNA, and we're all going to run a PCR, and the imposter's among us. So we're just going to compare the DNA of the imposter and see who it is. So here's the gel that we all run together. This is the imposter's gel on the left, the furthest to the left. So who do we think is the imposter? You may want to pause the video now and take some time to discuss. Okay, if you said that the imposter was orange, then you're right. Good job. So let's discuss a little bit and see how much we learned. So you may be thinking to yourself, why would we even do amplification at all? So why not just look at the DNA and see what size the gene is? So the answer to that is that it takes a lot of DNA to, a genome is a very big thing. So if you take someone's genome and you want to know just a target segment, you're going to need a lot of cells to do that. So that might be a whole arm worth of cells or a whole leg worth of cells and sequencing. And maybe you don't want to give up your arm or your leg. So what you might also be thinking is, what can we even use PCR for? What are other things that this is helpful with? And maybe why is this together with some cutting edge research being done? So I'm going to give you some pictures to look at, and those will be hints, and you'll have time to discuss with your group after each picture. So you're answering, what is another use for PCR? And this is your hint. Okay, so if you said you can use PCR to clone, then you're right. We can take cells and we can inject these cells into empty nuclei, and we can put them into a surrogate, and the surrogate can give us an exact clone of those cells. But first we have to amplify them, and we have to replicate them, and we do that with PCR. Okay, so next hint. Okay, I hope you all can recognize that one. So this is COVID-19 tests. So you may know, or maybe you've had a test, they'll do a throat swab or a nasal swab, and the bit of DNA that they have is not enough to compare to the COVID. And when they amplify your DNA, they're then able to compare it, and they can have a measurable way of knowing whether you have COVID-19 or not. So this one should be pretty easy. Okay, so if you said forensic science, or if you said identifying perpetrators or crime scene murderers, then you're right. So that is going to work exactly the way that we did it with our Among Us game. They're going to amplify the DNA that they find, and they're going to compare that to DNA that they got off of the crime scene and find exact matches, which will tell them if that's the person who did the crime or not. So this one may be a little bit harder. Okay, so if you guessed something along the lines of drug design, then you're right. So the way that PCR works here is we may know that there's a region of DNA that could be overactive, or it could be a cause of a disease. So if we know that, then we can run PCR, and we can design drugs that maybe inhibit that gene or somehow stop it. Okay, so take a second to guess what this one is. Okay, so if you guessed that it is a paternity test, then you're right. So paternity tests are done this way, and that's very similar to our Huntington's disease or our Among Us example. We're going to take the same region of DNA from a parent and then from the offspring, and you should be able to compare the DNA here. And that will tell you the size, and that will tell you that it's identical. So just a couple more. This will be a very hard one to guess based on the picture. Let's see what ideas you come up with. Okay, so this one is gene therapy, and we haven't talked a lot about how this is done or what other research PCR is being connected to, but if you've heard about CRISPR, then you know that gene editing is a tool that is pretty hot in the scientific world right now. So the way that gene therapy can work is you can take your gene of interest and you can cut it out of a diseased DNA, and then you can replace that DNA with healthy DNA and hopefully cure the disease. So I'll give you just a couple seconds, or you can pause and think about this one. Okay, so the evolution of a species is something that can be aided by the PCR technique. So you might be able to take regions of DNA and replicate them through PCR, and now you have billions of PCR from 2 different species, and you'll be able to compare those, especially if you sequence them, like we saw in an example of our base pairing. Okay, so last one, if you want to pause now and think about this one. Okay, so agriculture, this is a big one, and this happens all the time. So what's done is they may need to make a plant immune or resistant to certain bacteria or against insects, and the way that they do that is they take those genes, run them through PCR, amplify them, and then replace those genes with genes that are resistant to those things, and now you have crops that'll flourish. What they can also do is replace them with added nutrients and vitamins to make these more nutritious to us. So in conclusion, let's see what we learned about the steps of PCR. So after each of these questions, you may want to pause and see if you can get the right answer. What is the function of a primer? So the function of a primer is to frame our target DNA for replication. What are the 3 steps of PCR in one cycle? So the first step is denaturation. The next step is elongation. And the third step is annealing. So we're going to separate our DNA, we're going to build on our target DNA, and then we're going to re-anneal with twice as much as we had in the previous cycle. So how many times is a cycle of PCR typically repeated? So this will be millions and billions of times. Okay, and what are the 4 bases? If you know those or if you caught them. So in DNA, they're adenine, guanine, thymine, and cytosine. And I'll be really impressed if you can answer this last question. What invention won the 2020 Nobel Prize in Chemistry? So the answer is CRISPR-Cas9 editing technique. And that's it. So you guys are really awesome, and if you're interested in biology or medicine, looking forward, the University of Arizona has so many wonderful resources, and it's a great school to go to. So if you have any interest in this, that's where you should go, and keep studying and keep doing the best that you can do. Good luck.